Where do I go? Okay, I'm going to get started. Can everybody hear me okay? Great, great. So let's get started. So today is going to be the last lecture of the semester. So after today's lecture, there won't be any more live stream lectures. But every Monday until final exam, we do have the study session from 11:30 to 12:30. Okay. And if you have any more questions about homework, if you can't make it to the study session, you can always email me. But it's better for the study session because you can ask verbally, it's live interaction and stuff like that. So that's number one thing, today's last lecture. Number two is after today's lecture, I'm going to talk about the biofuels lab again. And that will help you to figure out what to do for the rest of the lab. What is the last thing I want to say? There was one more thing, which is, um, well, it will come to me later. Okay. So let's talk about, so last time we talked about glycolysis, pyruvate processing, and what is the last one? And Krebs cycle, right? So at that point, all the electrons between the carbon-carbon bond are all stolen or captured by electron carrier like NAD plus or FAD. So today we're going to talk about what happened after those first three steps. So you collect all these electrons now, and then where are you going to take them and how are you going to cash that in to become ATP, right? So far we only made, of the three steps, we only made 4 ATP, which is very little considering how much ATP cells use each second. Okay, so the next step is going to be electron transport chain. And what you need to know is, number one, the electron, this is mitochondria showing you in this picture. So electron transport chain is in the inner membrane. Remember there's an outer smooth membrane for mitochondria. And then there is an inner membrane in the mitochondria that's all folded up. Okay, so there, for electron transport chain ETC, there are four complex. So you see here the blue here, 1, 2, 3, 4. Okay, and you need to understand their function. Okay, number one, each one of these protein is a proton pump. That means it pump a hydrogen ion, H plus, every time when the electron get transferred from one to the other. Okay, and since it's a pump, that implies it's active transport. So it's pumping against concentration gradient. Um, and um, and if it's against concentration gradient, that means it has to be spending energy, right? And the energy that it gets is, so it gets its energy from um, transferring electrons. So when they transfer electron in the ETC chain, it can regenerate NAD and FAD, which is the electron carrier, so it can go back to the beginning of cellular respiration and start the whole process again. B is correct. This is the exact reason why the inner mitochondrial membrane is folded up so it can have more surface area to have a lot more ETC on it. Okay. So here is a picture of the ETC again. Now remember, this is the inner mitochondrial membrane right here. These green things here are ETC. So each one is a proton pump, right? So if you look at this ETC chain, the way it works is a chain, right? So you can imagine the electron will transfer from one complex to the other one like this. Now remember, the only reason the electron will move like this is because the number one guy. Okay, so let's start from the beginning. So NADH and FADH2 will donate its electron to the first ETC here. And the reason that they will donate their electron to the first ETC is because this ETC complex has a lot higher En than NADH or FADH2. So when these two molecules get close to ETC number 1, the electron is going to get stolen by ETC 1. Okay, so that's how it works. So same thing happened. ETC number 2 has a lot higher En electronegativity than ETC number 1. So that's why it grabs electron from this guy when they are right next to each other like this. So the electron always try and move like this. It always move to a complex that's way higher En. Same thing here. So you can imagine that complex 3 will have much stronger En than 2, so on and so forth. And then number 4 will be stronger than number 3. And then the last of all, if you see here oxygen, oxygen will be the strongest of all. It will have the highest En of all. So that's why it accepts the electrons at the final step. Okay. So what happens is as electron move from complex 1 to complex 2, because this is a higher En, so actually as it moves, uh, it will be attracted to this guy a lot stronger than this guy. So the energy is released because it's, remember we talked about how when you move to a more electronegativity molecule, a more electronegative molecule, the electron will lose its potential energy to move around. So when it moves from number 1 to number 2, some of the energy of the electron is going to be uh, lost, but that energy is actually being used by ETC number 1 to pump proton. Remember, it requires energy to pump, right? It's active. So the energy comes from moving the electron. So the energy difference between the electron in here and the electron in here, that energy difference is used to pump hydrogen ion. So that's what it's trying to say in this slide here. And then as it pump hydrogen ion, you see there's more hydrogen ion here. So it's building a proton, hydrogen ion, H plus is same as proton. So it's building a proton gradient in the inner membrane space. And I'll show you in a minute what it means. Now here is a picture of mitochondria. This is the outer membrane, right? This is the inner membrane that's all folded up, and that has all the electron transport chain protein here. So when it pumps proton, it pumps from matrix here to the inner membrane space, which is the tiny space between the outer membrane and the inner membrane here. See that? So then you build up a lot of hydrogen ion right around here, which is right between the first two layers, right? And that's the reason why in a minute I'll show you. So just remember when it pump hydrogen ion proton, it pump to the inner membrane space. So it's from matrix to the inner membrane space. So this is talking about when, I remember in the first step in this electron transport chain, when FADH and NADH donate its electron to ETC number 1, then they will become their empty electron carrier again, NAD plus or FAD plus. So then these empty electron carrier can go back to the beginning of cellular respiration to pick up more electron again. So the whole process can start. So one of the main purpose of ETC is to regenerate these electron carriers, to empty them in a sense so they can go back to the beginning of the process and start picking up the electrons again. So then once they pick up the electron, then they come right here and dump the electrons to the first ETC. Okay, so that's first. And as I talked about it last slide, how each one of these ETC is getting higher and higher En, and at the end, oxygen has the highest En of all. So the electron will move from all of this, and then at the end will end up to be with oxygen. And when it end up with oxygen, then it become water. So without oxygen, sometimes you don't have oxygen, right? Without oxygen, this transfer of electron will stuck. So because every ETC complex can only hold like 2 electrons or so, right? So if you can unload the electrons from ETC4 to oxygen, then ETC4 is not available to pick up any more electron, and then this guy will back up to, and then eventually back up all the way to the number 1 complex. In that case, then the NADH cannot donate its electron because this guy is already full. So that's why oxygen is so important, because you need that to keep this chain going, right? You have to keep emptying the electron to the oxygen to become water so it can go away. And then other oxygen come in and pick up the electron. If not, this whole chain is going to get backed up. You can't donate the electron to it anymore. So there's no regeneration of NAD plus. So the whole cellular respiration reaction is stopped, because remember the first step is glycolysis. And one of the input for glycolysis is NAD plus, because you need to go in and pick up the electron, right? Without NAD plus, this whole cellular respiration cannot be possible. Yep. B is correct. So oxygen needs to be at the end of the ETC number 4 protein to accept all the electrons, right? So for this part of the chapter, you guys need to be able to trace the electrons, how it moves, right? From what to start with and end up in where. So for now, you will know that it start all in NADH or FADH2. That's where all the electrons originated from, right? And where do those guys get the electrons from? It's from the first three steps, from breaking down the carbon-carbon bond. Okay, so then it moved to the first ETC complex, and then second, and third, and then fourth, and at the end go to oxygen. Okay, let's try this question now. Yep, right on is mitochondria. If you don't need oxygen, you don't need mitochondria. Now, so far you know that most of the energy that harvests from breaking down glucose, the electrons, right, we're talking about, have been used to pump protons to the mitochondria inner membrane space, right? As the electrons move from one ETC to the other, the ETC is proton pump anyway, so it pumps proton to the inner membrane space. So a proton gradient has been built, but why? Why are you spending so much energy building a proton gradient for what? Now, proton gradient, just to be clear, is potential energy, right? So here is a few terms you need to understand. So the reason to build a proton gradient is so that the protons want to move back into the matrix. And when it moves back into the matrix, it will generate ATP. And this way of generating ATP is called chemoosmosis. Now, you guys already know the word osmosis, right? So what is chemi here means? It's chemical. What kind of chemical are they looking at? They're looking at hydrogen ion, okay? So the moving of hydrogen ion by osmosis, high concentration to low concentration, right, will generate ATP, and this is what is called chemiosmosis, generating ATP by moving hydrogen ion. And proton want to come back into the matrix by two different forces. One is the concentration differences, right? There's high concentration in the inner membrane space, and there's lower concentration in the matrix. That's one force. Now, when you have high concentration of hydrogen ion in the intermembrane space, then there's a charge difference, right? It'll be more positive in the intermembrane space versus the matrix space. So the charge differences will also attract the proton to come in. Now here is what it looks like. Now here is the inner membrane space, right? Inner membrane space, and then this here is the inner membrane. This here is the matrix. So remember the ETC, let's just imagine the ETC around here. They keep pumping proton to this tiny space here, okay? So there's high concentration of hydrogen ion here, and then there's lower here. So hydrogen ion want to come back in because it's higher concentration outside than inside here. And plus, when you look at all these positive here, there's a positive charge here, and there's a relatively negative charge here. So positive is attracted to negative, so it wants to come in. But remember, hydrogen ion, even though it's small, is charged, so it cannot cross the membrane freely. It has to have some help, right? So it turned out there is a special protein called ATP synthase that can allow the hydrogen ion to come through. But every time when the hydrogen ion go through this particular channel protein, it happens to be ATP synthase, an enzyme, it will make it spin really fast. So this is why it's talking about ATP synthase is a rotary motor. So when hydrogen ion enter through this channel, it will spin really fast. Now look closely. It's going go in, and then spin. See that? So how fast does it spin? It spins about 350 revolution per second. Now you guys have no idea how fast that is. So it spins 350 round per second. So let me give you a scenario. When you drive your car, there's like a speed meter right in the center, and then there's another one, say RPM on the side. That RPM is telling you how fast your engine is spinning to give you a certain speed. Okay, so let's say if you're driving 40 miles per hour, 40 miles per hour, your engine is spinning about 2,000 revolution per minute. Now let me do my little calculator and tell you how fast is per second here. 350 times 60. So when you're spinning as fast as this ATP synthase, you're actually going 21,000 revolution per minute. Now compared to 2,000 per minute for a car driving at a 40 miles per hour, this is 10 times as fast. So at that speed with the ATP synthase, it's as if you're driving 400 miles per hour that fast. So can you see how fast this guy is spinning now? It's like going 400 miles per hour, which is pretty amazing. So at this point, when hydrogen ion go through here, it spins that fast. It's using that mechanical energy of spinning, putting the phosphate onto ADP and turn it into ATP. And you can imagine this could be pretty fast because it doesn't require a substrate or anything like that. Just like in the substrate level phosphorylation, right? So as it spin, it put the phosphate onto ADP and then spit out ATP. So this way of making ATP is really fast. And you can make a lot of ATP from it. So that's why most of the ATP is produced by this ATP synthase by spinning really fast. And the way that it will only spin when there's a hydrogen ion moving through it. So remember, it's a channel protein, but also it's an enzyme. So producing ATP by chemiosmosis, we talk about it. Chemiosmosis means that the movement of hydrogen ion to create ATP, right? It's called oxidative phosphorylation. Okay, and the reason, well, you know the word phosphorylation, it just means that turning ADP into ATP because you're adding a phosphate group to it, right? Phosphorylation, you should know that word already. Now, what is oxidative means? Oxidative means what? Oxidation means a loss of electron, right? Who is losing electron here? FADH and NADH. Remember, they lose their electron to ETC1 to allow this proton gradient to be built. So then you can have this ATP production. So that's why making ATP in ETC is called oxidative phosphorylation because it's involving electron moving along. And the first step is that the electron carrier has to donate the electrons. So to put all these into an analogy, think about if you have ever visited a dam, right? A cement wall somewhere, you know, they put all the water, they pump all the water behind the dam. So a lot of these dams will have hydroelectric turbine. So it's not a complete 100% cement wall. There's like little doors there. There's like turbine there and stuff like that. So you can imagine ETC is a bunch of water pump to keep the water behind the dam, right? And then ATP synthase are those little tiny door that are turbines. And when the when they need electricity in the dam, they will let some of the water behind the dam come in through those doors, and those door has turbines. So it will spin those turbines really fast and make electricity that way. So the same way, so if you build up this hydrogen ion concentration behind the inner membrane, when it come in through this ATP synthase, it will generate energy in that way. So chemiosmosis, the analogy is the movement of water through the turbine, generate electricity. In this case, is the movement of hydrogen ion through ATP synthase to generate ATP. So can you kind of see that? That's how it works. So that's how your body convert all these electrons from carbon-carbon bond into potential energy, which is proton gradient between the inner and the outer membrane in the mitochondria. And when the hydrogen ion comes back into the matrix, then it will generate ATP, and that's it, in that way. Now, is there any question? Let's try this question then. So what is the direct energy source to make ATP synthesis? Yep, C is correct. You're tapping into the potential energy. Remember, it's the hydrogen ion concentration differences. So C is correct. So we're not doing this because you guys can you do this? You want to do this? Maybe we can do a little bit of this just to be sure you're getting it. Let me change it to a pen. So we have what is same between the two. Let's say we have oxidative phosphorylation here. We have substrate level phosphorylation. Now I think I only see 6 of you here. So let's everybody give me something that is either the same between the two or differences between the two. Okay. And I'll put it down for you guys. Okay, I'm gonna write that down. So Dahlia say oxidative phosphorylation involve chemiosmosis. That's very good. There's actually a lot of differences and there's also some in common. So come on, you guys, everybody give me one thing and I'll fill out the rest. What is in common? Yep, Harley said that SLP only make 4 ATP, so 4 ATP it makes small amount of ATP, right? We haven't talked about how many ATP does oxidative phosphorylation make. And Neri said this is in glycolysis. Very good. Okay, and Neri filled that out already. So oxidase for phosphorylation make lots of ATP. Let's just put down 38 ATP. Very good, Heath. They both happen in the mitochondria. This is, so N has a lot of good stuff here. So N said you need a proton gradient here, right? Proton gradient. Here you just need, you use substrate, right? Enzyme substrate reaction. Hope you guys can read this. So it's substrate enzyme reaction. Remember how it transferred the phosphate group from one substrate to another? Let's see. Misty, do you have one? There's so many of them. Okay, let's try this. So Aneri said that SIP, substrate level phosphorylation, SLP is in glycolysis. But in theory, if you remember, actually it's in all three other process, all first three, right? In glycolysis, in pyruvate processing, in Krebs cycle. So the first three steps in cellular respiration all use SLP, substrate level phosphorylation, to make ATP. Okay. Yeah, Misty, you got the last one. So ATP is made by both here, right? So you have a lot less ATP making in SLP than in oxidative phosphorylation. And like I said, this is a substrate enzyme reaction, and this is, like Ian said, is a mechanical energy. Now the enzyme they use here is called ATP synthase, right? You guys can write that down. And you can also write down this also use ETC, right, to transfer electron. And what else? Yeah, I think that's the most of it. So can you see that? What is in common is they're all in mitochondria. Oh, we can write down here, this is in the inner membrane, right, of mitochondria. Even though they're both in mitochondria, their location is a little different. So inner membrane, this is where. Now, glycolysis is in cytoplasm. So SLP can happen in cytoplasm. And the other two step is in matrix, right? Mitochondria. You know, I'm writing like a kindergartner again. Mitochondria matrix. Can you see the differences now? The location is different. The enzyme that they use is different. Um, involve ETC, proton gradient, chemiosmosis, make tons of ATP. I'm going to give you a minute to write that down if you want to. If no, get a drink of water. Because when I move away from this slide, all these will be erased. If you're done writing, can you just send me an okay or yay or something so I can move away from this slide? Good, good, good. I'm gonna move. He's just very smart. He took a screenshot. That's very good. That'll be easier and faster than writing it down. Okay, so some of you already got into this slide, knowing that substrate level phosphorylation only make 4 ATP, very little, and most of the ATP is made in this last step in ETC. We can do this, but if you look at any of these compounds, you already know that arsenic is poisonous, right? And carbon monoxide is also poisonous. This is how people kill themselves when they have the car running in the garage. Remember that? So all these stuff is poisonous to you only because it messes with the cellular respiration pathway. For example, if you look at carbon monoxide, it binds to the hemoglobin, which is a protein in red blood cell that transport oxygen. So in the case of carbon monoxide poisoning, the hemoglobin is occupied by carbon monoxide, so it cannot carry oxygen in that sense. So you're out of oxygen, right? Without oxygen, the ETC is not going to work. Agree? Now, arsenic is another poison that recently was shown in chicken, in rice, in all kinds of stuff. And the reason it's toxic is because it inhibits pyruvate dehydrogenase, which is the first step in pyruvate processing to change pyruvate into acetyl-CoA. So it kind of mess up the second step of cellular respiration. So if without acetyl-CoA, you cannot have any Krebs cycle going on. Remember, Krebs cycle is the one that generates the most reduced electron carriers like NADH, FADH2. So that explains why arsenic is very toxic. Now, some of these are antibiotics, right? You know the antibiotic is used to kill bacteria, but do you understand some of the antibiotic also kill regular human cells because it depends on how it works. Like some of these, it work on, like oligomycin work on ATP synthase, right? So then the hydrogen ion cannot get through it. So then there's no ATP being made. And this guy, antimycin, they bind to ETC number 3 so that it cannot transfer electron. Okay, so this mess up ETC, this mess up ATP synthase. Cyanide is then also another poison. Now this is also messing up ETC. So then there's no electron get transferred to oxygen. So a lot of these poison um, work on blocking or destroying cellular respiration pathway. So you cannot make ATP anymore. Then you will run out of ATP very soon and die. Now, DNP is a very interesting chemical. It used to go by there used to be a diet drug called fen, and the active ingredient is DNP, and DNP work by destroying the proton gradient. So the way it works is it insert itself in the inner mitochondrial membrane. It's kind of like a channel or a hole. You can say pores or holes that it makes holes in the inner mitochondrial membrane. So when proton, you know how the ETC keep pumping proton to the intermembrane space, and it can only come through ATP synthase. But now with this chemical in the inner membrane, it can go through these holes that this chemical makes. So it's in a sense it destroys proton gradient. So when there's no proton gradient, what happens? Then you don't make any ATP. So people, of course, on this drug is losing weight because they're not making ATP, right? But at the same time, they're dying. They don't even know that. So I guess there's a huge price to pay for being pretty. Ah, okay. So here's a summary about all we talk about. So all three steps in all first three steps here, the glycolysis, the Krebs cycle, the pyruvate processing, is to extract all the electrons from glucose, which at the end, see this NADH here, NADH here, FAD, all these is going to get carried by electron carriers and all feed into this last step here we just talked about today, the ETC chain. And through ETC chain, you make a ton of ATP, of course, here. You regenerate NAD plus and FAD. So these two guys can go back to each one of these steps to start carrying electrons again. Um, just remember our body has limited supply of NAD plus and FAD plus, so we just have to keep regenerating it, keep reusing the same pool that we have, okay? And of course, at the same time, oxygen is turned into water, right? And this water can be used in the other biological process, like in hydrolysis and stuff like that. So nothing is gone to waste. And hopefully at this point you understand oxygen that we need so badly every second of our life, right? We breathe constantly to get oxygen. Is because this whole process, especially this part here, require oxygen. Without oxygen, this whole chain is stopped. So you cannot get ATP. Now remember we talk about input and output. Let's try this now. What is in the next slide? No. What is the input for ETC, the last step? What goes in? Let's try this. Now you guys answer me with your text. What is the input for ETC? And you know, my next question will be what are the outputs? So you should tell me what are the outputs for ETC. And then the next question is what are the what are the inputs for ETC right now? And then next question will be what are the outputs for ETC? Let's try input first. What goes into ETC? So input in and heat has, of course, you input, you have to put, you have to have NADH, right? FADH2. Good. Oxygen, of course, is input. There's one more thing is missing. There's one more input. See if anyone can come up with that. Can anybody think of the last input? Look at this. If this is an output, which is ATP, what needs to go in? And there is close. It's not ATP. ATP doesn't go in. It's ADP, right? ADP will go in here and turn into ATP. And now hydrogen ion is not what I'm going after. But what I'm trying to say, okay, so let's summarize. So the inputs for ETC, yeah, is oxygen, NADH, FADH2, and ADP. Now, people tell me what are the outputs for ETC? Come on, tell me what are the outputs for ETC? What comes out at the end of ETC? He's very good job. So that's exactly, you come out. So remember, every input will need to be accounted for as an output, right? So FAD, NAD, ATP, and water. Very good. Okay, so is there any question about this right now? We're gonna take some questions for this whole ETC because next two slides should be easy, and then we're gonna go into fermentation. So let's take 5 minutes break. If you have question, will you type it? And after the break, I will answer those questions. You pull up the worksheet for today. Okay, let's get started again. So I assume there's no question, nobody typed anything, so let's go on. So what happened is, hopefully you guys don't just eat sugar, right? Remember that we start with glucose in cellular respiration. What if you eat other stuff like fats or protein? How would that fit into cellular respiration? So if you look at this picture here, and the laser pointer. Of course, if you eat carb, starch, sugary stuff, even like vegetables and stuff, eventually they all turn into sugar, right? You break it down into the monomers. Most of the time, it's going to be glucose. So there's no problem. You feed straight into the cellular respiration pathway. But what if you eat fats? So fats need to be processed with enzymes in your body to break it down into glycerol. Remember, there's two components for fats. You have the fatty acids, tau, and the glycerol. They break that apart and then modify them so the glycerol can fit into the beginning of pyruvate processing. Fatty acids can get modified and go straight to become acetyl-CoA, which then goes straight into Krebs cycle. Now, protein, remember there's amino acid. So they will take away the amino group and then can turn amino acid into acetyl-CoA and then off into Krebs cycle. So one way or the other, the fats and proteins you eat will get modified and then fit into different steps in cellular respiration. So you can kind of tell why your body likes sugar so much, because they start from the beginning, go all the way to the end without much processing, right? So, but fats and proteins will take longer. That's why a carb is so much faster to digest and give you energy. Fats and protein, on the other hand, needs to be modified before it can fit into cellular respiration. Okay, so you are just like bacteria. Remember, bacteria prefer glucose. For ATP production, we as humans prefer carb first. Then we'll do fats and proteins. So remember those diet about, well, Atkins diet, the one earlier, and then now it's really popular is keto, and then of course it's a low carb diet and all that. Now all that diet will help you lose weight because they don't they limit the amount of carbs that you eat, and they focus on fats or proteins, right? Um, so your body has to work harder to extract energy from these guys. Um, and sugar, a lot of time, excess sugar will turn into fats in the body. That's why not eating much carb will help you to lose weight, but you cannot go without carbs because you still need the vegetables, the fiber, the vitamin, and all that stuff in it. So I wouldn't recommend a zero carb diet. But cutting down your sugar and starch is going to help you. Okay, Ian is asking, can glycerol turn into pyruvate? I don't think it's straight up like this. So glycerol, if you look at this, it will go into not go straight to become pyruvate. If it would, it will go like this. The arrow will go straight. It needs to be modified a little bit before it becomes pyruvate. Now, what if you have too much, right? So let's say you have all of a sudden, you have too much sugar in your body, or uh, your cellular respiration is just overrun in a sense with all these products, right? So if you have too much sugar, of course your body is going to turn into fat or turn into glycogen to store in the liver. If you have too much pyruvate, right, at the end of glycolysis, they can turn pyruvate into glucose again if they don't need it. Acetyl-CoA, if you have too much, after the second step, they can recombine and make it into fatty acids, right? Turn it into fats or phospholipid. If you have too much Krebs cycle stuff, they can turn all that into amino acid. So you see that when you have extra, let's say you already have extra and then your cellular respiration stop, all these extra substrate or products from each step can be turned into useful stuff that your body can use later. Okay, so we are done with cellular respiration. Now remember, cellular respiration only happens when there's oxygen, right? Because the ETC has to work with the oxygen. But what if there's no oxygen? So let's talk about what if there's no oxygen. And this part here, fermentation, is part of your biofuels, because biofuel lab using fermentation to turn sugar, glucose, into alcohol. So you learned this today and you'll be all good for the biofuels lab. So remember, in glycolysis, you start with 6 carbon here, right? And then after glycolysis, you split it up into 2 molecules of pyruvate. Each one is 3 carbon. Now, if oxygen is there, then go into, of course, mitochondria, go through all the rest of the steps. But what if oxygen is not available? So let's take a look. If oxygen is not available, then you have a problem, right? The problem is you don't have enough NAD plus around to extract electrons from glucose. So in cellular respiration, NAD plus is regenerate during the ETC step. Without oxygen, the ETC step doesn't work, so you can't generate NAD plus, but there's a way to regenerate ETC, to regenerate NAD plus. So the purpose of fermentation is to regenerate NAD plus from NADH. Okay, and let's talk about it. And there's no ATP being made. So in this case, I always tell my student, fermentation is kind of like when you don't have oxygen at times, you're buying time, you're waiting for oxygen to kick in. So you just kind of like try to buy time to make some NAD plus, and you're not making ATP just to keep the things going. Wait till the oxygen comes back in. Now, fermentation is in the cytoplasm, just like where glycolysis is. So think about it this way. After glycolysis, which is in the cytoplasm, if there's oxygen, it goes into mitochondria. If there's no oxygen, the reaction will stay in the cytoplasm. Okay. So there's 2 types of fermentation. One is alcohol, because it makes alcohol, fermentation. And one is lactic acid, same thing. This fermentation reaction makes lactic acid. Okay, so here's glycolysis. With oxygen, it go into mitochondria. Without oxygen, it stay outside in the cytoplasm to go through fermentation. Now let's talk about alcohol fermentation first. Now in biofuels lab, we use yeast to do this fermentation because what happened is, remember pyruvate at the end of glycolysis supposed to go into mitochondria, but there's no oxygen, so it's not going into mitochondria, right? And at the end of glycolysis, you have pyruvate, you generate some NADH, correct? So in order to, because there's no oxygen, in order to regenerate NAD plus, what happened is this NADH would dump its electron to pyruvate. Let me draw an arrow here. Because just remember there's no oxygen. So this NADH cannot donate its electron to the ETC because there's no oxygen now, right, in fermentation. So what happened is the electron in this NADH will donate to pyruvate. Okay, so when this reaction happens, you get alcohol, you get your CO2. That's why your beer is so bubbly. Okay, now you also get NAD plus. See that? By dumping the electron to pyruvate, you regenerate NAD plus without oxygen. But that's very little amount you regenerate. So like I say, it's buying time, waiting for the oxygen to come back. So then this NAD plus can then go back to glycolysis here to start glycolysis again, right? Because glycolysis, one of the input is NAD plus. Now, so if you look at this here, you have glucose going through glycolysis, generating ATP, right? Generating NADH, right? Because the electron here would donate to NAD plus. So that's glycolysis, this part, and then you generate pyruvate. And then right after pyruvate, with no oxygen, the pyruvate is going to get combined with the electron with NADH. Then we generate NAD plus, right? And this guy here, the arrow is showing that it go back to this. So in that case, so then glycolysis can happen again. So in fermentation, which is this step, you generate no ATP. But since glycolysis can keep happening, so you will generate tiny bits of ATP because of fermentation, allowing this NAD plus to go back. If not, you got no NAD plus. So that's anaerobic cellular respiration without oxygen. Okay, so like I said, it's just a way to buy time to allow glycolysis to go on to generate a little bit ATP so the whole chain doesn't have to stop. Now, if you think fermentation is a new thing, it's not. Human has been using fermentation for ages. If you like yogurt, yogurt is a product of fermentation. Cheese, bread, we put yeast in it, remember, to make bread. Of course, beer, wine, and stuff like this. Soy sauce is also a fermentation product, vinegar. So all these stuff that we use on a daily basis is from fermentation. Now, let's see. Now we're going to talk about lactic acid fermentation. Now remember this fermentation generate lactic acid. That's why it's called lactic acid, right? So of course this happens when there's no oxygen around or there's not enough oxygen go around. I should say that in fermentation usually it's not a complete absence of oxygen. It's the lack of, there's not enough oxygen to go around, okay? For example, if you look at this guy swimming really hard, so in our muscle, in human muscle here, when we exercise really hard, the demand for oxygen is too high, so our body cannot supply oxygen to the muscle fast enough. So at that point, the muscle will go through lactic acid fermentation because the muscle is just using up too much oxygen. Now here's the equation for it, and I think in the worksheet it asks you to make sure you write down the input and the output in the worksheet, right? So you start with the same thing. You have the product from glycolysis, which is pyruvate NADH. Same thing happened. NADH would dump its electron to pyruvate. So now you regenerate NAD plus. But at the same time, because this reaction happened in muscle, so it will have lactic acid. Okay. Now here's the same thing. It's the same picture showing you that how fermentation and glycolysis happen at the same time. So because of glycolysis, you generate pyruvate, ATP, NADH, right? And without oxygen, this pyruvate will react with NADH, regenerate this NAD plus, and then this NAD plus go back to glycolysis. So then this glycolysis can go on again, and then you have lactic acid. So if you ever exercise at all, you will understand when you work your muscle too hard, your muscles start feeling kind of what? Pain? Burning? Yeah. That's how you know your muscle is getting weak, and it's because of the building up of lactic acid. So that's why you have those sensations. So it's a good time to start if you have those feelings in your muscles. Now, as you see that I already said that fermentation is extremely inefficient. So if there's a choice, organism always prefer cellular respiration over fermentation because fermentation doesn't make much ATP except the two in glycolysis, right? So this is a comparison. You see that in fermentation, you only make 2 ATP through glycolysis. Fermentation itself does not make any ATP. But the goal is this here. You're regenerating NAD+, so then glycolysis can continue. Remember, this is comparing to cellular respiration. You're making, what, 38 ATP for one glucose. In fermentation, you're only making 2 ATP per glucose. Let's try this question. Good, everyone C is correct. So the goal is to regenerate NAD+, right? So we're done, but I want to talk about, let's look at the worksheet first. You know the thing I forgot in the beginning? I was mentioning today, this one is the last lecture. And if you have questions, you can email me or come to the Monday 11:30 study session. I still have those until the final exam, which is May 11th. And about the final exam, that's what I forgot. So the final exam is going to be the same format as all the other exam. Open book, open note, online. You're going to take it in D2L. I'm going to give you 2 hours for it. And this time, it's going to be 50 multiple choice. It's no essay anymore. And I focus the questions that is going to be on the final exam is on the big stuff. For example, it's like stuff I want you to remember a year from now. So it's the big concepts. So in regular exam, I test you guys on all the details I talk about, right, in class. But in final exam, my goal is not about all these details. I want you to remember all the big ideas that we talk about. Hopefully you take away something that can be with you for the rest of your academic career. So it's 50 multiple choice questions, all about the big stuff. Of course, if you know the smaller stuff, it will help you with the big stuff. So it should be pretty easy compared to regular exam, and hopefully it will bump your grades up a little bit. And I will have it open up later on, maybe a week before final exam. Now, final exam is going to be on May 11th, if I'm correct. That's the last day of the semester. So I'll have it until May 11, 11:30 at night, same thing, 2 hours. So you can do the final exam that way. If you have question, it's a final exams is comprehensive. So it covers chapter 1 to cellular respiration, all the chapters. Okay. So let's look at the worksheet. If you have question, you can type it up and tell me. Let's look at the worksheet for today. Let's make sure there's no question about it first. And then we'll look at the biofields, okay? I don't think you need to draw in this worksheet, so you guys can type it and then email it back to me. Okay, the first one, if you guys have your worksheet, now of course you know what A is supposed to be. It's looking at this thing here, right? A in glycolysis become B. So you should know all of that, and then B will go into the mitochondria. That's assuming there is oxygen, right? Um, and then go into mitochondria, and then will at the end will fit into Krebs cycle. But what I want to point out is this. Let's see, whatever. So you guys always get confused about D. Okay, D and what else? Oh, D and E. Now the hint I'm going to give you is this. Now both D and E is the inputs for ETC here, right? But D only comes from Krebs cycle. Can you see that? But E comes from Krebs cycle, comes from pyruvate processing, and also comes from glycolysis. So just look at all these arrows. So E comes from glycolysis, comes from pyruvate processing, also comes from Krebs cycle. And yet D only comes from Krebs cycle. So now there's a big hint and you should be able to do that. So that is the one big thing. Now this question here, number 8, is the same as the other worksheet. If it's incorrect, you underline the word that is incorrect and fix it. If it's correct, you just write true. And that's all you have to do. Now, the one that I want you guys to look at is C here. It said proton or hydrogen ion is pumped against its concentration gradient, which looks good so far, right? But from where to where? This is the key part. Is this correct? A lot of you missed this. So you need to make sure you understand where does it pump. And you go back to this PowerPoint here. Remember, I have this little picture here. Hydrogen ion pump from where to where? Here's where you find the answer, okay? Now, one thing I want to point out is this. Do you remember the overall equation for cellular respiration? But in the first 3 steps, let me show here. In the first 3 steps of cellular respiration, the overall equation is what? Glucose plus oxygen, right? Will turn out to be CO2, H2O, and ATP. Now, if you look at this first 3 steps in cellular respiration, you don't see any oxygen, correct? You see glucose here, but there's no oxygen here. There's no oxygen here. There's no oxygen. Oxygen only appear in the ETC chain. Okay, so the overall equation for cellular respiration reflect the summary of all 4 of these reactions, not just one step of this reaction. So you see that glucose is from here, right, in the equation. Oxygen is from here, and then it becomes CO2 is is from here and here, right? Water is from here, and of course ATP is from all these together, right? Can you see that? I just want to point out that the equation is a summary of all four of the reaction. It doesn't just show one, all four of them. That's why you will see oxygen, even though oxygen is not in the first three steps. Okay, let's see another question. This is the same kind of thing, talking about fermentation, right? Now this here, bubble, you need to remember in alcoholic fermentation, what is the product? Let me show you. So I have an equation, nice equation here, right? In making alcohol, you have alcohol, and then you have your CO2 here. So this is what the bubble is. Okay, the function, and here inputs and outputs, remember to do this kind of equation here. That will give you inputs and output for alcohol, and of course for lactic acid, it's the same thing, right? Remember, this is what I'm looking for, very detailed inputs and outputs. So that should be good. Everything else shouldn't be a problem. Okay, ABC on number 9. What is number 9? Okay, so it's about this picture in, right? A, so let's look at what is A. So now on this side, it's talking about alcoholic fermentation, right? Because it's yeast. On this side, it's talking about lactic acid fermentation because it's muscle. Now, what is released in A? Now, the way to look at this is in this alcoholic fermentation, you generate something, right, that will go through here and turn out to be this. You're asking about A, B, and C. B is already given. So this B molecule is already given, acetaldehyde. Okay, so you don't have to worry about B. But what is released during alcoholic fermentation? What do you think? What is one of the... So we only have a few things going on, right? So we have in alcoholic fermentation, we have pyruvate, which is the which is this B thing, okay? Because pyruvate will turn into this acetaldehyde thing. So in here you have NADH, alcohol, CO2, and so these are the few things you need to pay attention to because this is already in the picture. So look at this, pyruvate is already in the picture. So this is already given. So what is being released here? So we have, so far we have CO2, we have alcohol, we have NAD plus and NADH and all that stuff, remember? So what do you think A is? Yep. So so A here is CO2, C here is alcohol. But what about D here? What do you think go in here and then come out as E and then go back into glycolysis? So what here react with pyruvate and turn it into CO2 and alcohol? So what do you think D is? Yep. So this is D is NADH will go into fermentation, come back out as E, which is NAD plus. Then it can work as an input for glycolysis. Then it come out as NADH. Yeah, you guys good, right? And same thing. See, this NADH will go into the muscle, work with the pyruvate, and then turn it into, I'm sure you know what this is lactic acid, right? And then go back into NAD+. Now, any more questions about this worksheet? If not, we go into the biofuels now. I need to open up the option. Okay, so let's go back to the biofuels. Hopefully, you guys done the soft chalk, right? And if you didn't come to the biofuel lecture PowerPoint, you can go back to listen to the live stream lecture. And last time when we were talking about biofuels, I said that you guys need to do the first 3 questions on the data sheet. Okay, so you need to, and all these, the first one you can find the answer from the PowerPoint. What is cellulose? Um, let me finish this, and I'm going to talk about the report. So let's talk about the design, and I'm going to talk about the report in a minute. Okay. So hopefully you already, so you look for the answer for cellulose in the PowerPoint. You should know that. And then this is really straightforward. Some of you already did that, right? To figure out what the plant biomass is supposed to be. And then you're supposed to research these enzymes, what their substrates are, and what the products become. And why do you care about substrate? Number one, you need to match the biomass you want with the enzyme you want, right? If the enzyme work with the substrate that is not in your biomass, then you probably don't want to pick that enzyme. Does that make sense? And the product will tell you whether because your goal is to generate the most amount of alcohol and you need glucose, correct? So you need to figure out whether the product at the end of this enzyme reaction will be glucose or not. If it's not glucose, it's not probably going to help you much. So that's why you need to know the substrate and the products for each one of these enzymes so you can make a good decision about what enzymes you want to use and what um, biomass you want to use. So you should do these 3 first, right? After you do these 3 questions, then I already posed this biofuel options on D2L in the biofuels fol 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 folder. Okay, so option 1, we have the group was using 2 different biomass, corn cob or corn husk, right? And they use only 1 enzyme. And then the second group used grapefruit peel. They use 2 different enzymes to compare. Third group, orange peel, used 2 different enzymes, right? Fourth group, coffee grind, 2 different enzymes. Fifth group or fifth option, they use potato skins, and then they have this 2 different enzymes. Okay, so after this 3 questions, you want to do some research on these options. See which one will make most sense to you, that will make the most alcohol. Okay, and then after you pick your option, where are you? What you're going to do is you're going to start with question 4. The goal of this lab is to make the most bioethanol, right? Since you don't have a lab group, you're going to be on your own. You need to think about a question. Let's say you're going to pick option 1, right? And you need to write down what your research question is, because without a question, you cannot have hypothesis. So for option 1, I'll give you an example. For option 1, the question you can have any question, but the question would be, for me, it would be something like, does corn cob or corn husk make more bioethanol with cellulase? So that's the kind of question I'll have. So if let's say I'm using option 1, my question for this lab will be, does corn cob or husk make the most bioethanol with cellulase. Okay, and then your hypothesis will be something like this. You have to, since it's either corn cob or husk, you will say, oh, I think corn husk make more because. You have to say because. Okay. Um, and of course, then in this question, you need to write down which option you use. Like biomass, for biomass, I'll write down corn cob, and corn husk. And now I'll write my enzyme of choice is cellulase, right? So in the hypothesis, you can say, I think corn cob will make more bioethanol because I'm just making it up. I don't know. You need to do some Google's research on it. So I'll say corn cob makes more bioethanol because it has more cellulose, which can react with cellulase and make more glucose, right? And that reasoning in your hypothesis can also come down here. So you're kind of writing the same thing, except in more detail. So in here, you can just write in one sentence in your hypothesis. Here, you can explain the research, the information you find from your research in more detail in a few sentences. Okay, does that make sense? So after you do that, you'll make a prediction about what you expect. So let's say my prediction is corn cob will make more uh, glucose because it has more cellulose, and therefore it will make more alcohol. The glucose will ferment into alcohol. Okay, so that's the prediction you have. Now, what will be your control group? So here's the thing I want to talk about. For your biofuel experiment, you need to have a control group. I'm going to have a new slide up here so I can draw. Now, I'm still using the same option. So I have cob, remember, versus husk. And I'm using cellulase as my enzyme. So the way to set up this is this. You will need, of course, you have tubes, right? Just pretend you're in the lab. So you have your biomass in here. Let's say that this tube here is corn cob. This here is husk. Okay, so you have cellulase, right? So what happened is for each one of these biomass, you need a control with no enzyme. So your control, the one with no enzyme, will be your control group. Why? Because you want to know whether your enzyme works, right? There is always some sugar without enzymes working, right? Especially if you're using like orange peel, some of these fruit stuff, there's always some sugar hanging around as a background, right? So you want to have a control with no enzyme to see what the background sugar is like. So for this one will be the one with enzyme cellulase. So that way you can compare this with this. See the sugar you generate after you do enzyme digestion compared to without enzyme, then you know how much it generated from the hydrolysis, right? Same thing here, husk, you have the enzyme here, right? And then you have no enzyme here as your control. So that's what that picture is asking you about. What is your control group? So in my case, my control group will be biomass with no enzyme. Serve as a baseline to compare with enzyme. Now, some of the other options, they have one kind of biomass. For example, I remember there's coffee, right? If you're doing just one single biomass, but two different kind of enzyme, I don't remember enzyme 1, E1, and then there's E2 to compare. First of all, the question for this kind of scenario will be, you can't say, oh, does this make more than this? But it'll be the same kind of question, except you will say, does this enzyme will make more bioethanol than this enzyme when it react with coffee? You see that how I changed the question a little bit, shifted to the enzyme instead of the biomass, because I have two different enzymes to compare. This one here, I'm comparing two different biomass, right? So in this case, you have for coffee and two different enzymes, you have the same thing. You have one with coffee, right? Two with coffee, three with coffee. So the control is always the one with coffee, but no enzyme. It's always the same. Every time when there's a control, it's always going to be no enzyme with the biomass. Now then you can have your enzyme number 1 here and enzyme number 2 here, right? So then you can compare what you get from this digestion to the baseline and this digestion from the baseline. Does that make sense? So let me answer some of these questions first. So Harley, you look at the options, you pick options like what I show on the work file. Okay, so if you pick option 1, then you are looking at two different biomass, and the rest of the option is looking at one biomass and looking at different enzymes options. Okay, so it depends on which option you pick, then you can either work with different biomass or different enzymes. So in the prediction is different because in hypothesis, you are, remember, it's educated guess. So you have a question before you, and then you're going to answer the question based on what you know. So it's not a prediction. Prediction is after the hypothesis. So let's say I wonder, like the question I have is, I wonder whether corn cob or corn husk make more bioethanol, right? My hypothesis is, I think corn cob will make more because then you have a little reasoning, and then the prediction is, oh, I see why you're confused now. Because in this scenario, it's very similar. Because your question is whether it will make more, right? Um, let's find another one. Yeah, in this scenario, they're very similar. So you can say the predict. Oh, let's see. That's how to make it a little different. So the hypothesis. Okay, you say, I wonder which one make more bioethanol, and you say, okay, I think corn cob will make more because it has more cellulose. Let's say that's what you're going to say. And then your prediction is corn cob will make more glucose because it has more cellulose, and that would turn out to be more bioethanol. Yeah, they're very similar, so don't worry about it. You can write similar stuff down in both of them. But in general, prediction comes after hypothesis. Okay, so are we clear about the control group? That's the part that you need to know. So the control group is always the one without enzyme, okay? And then because you need to compare with the one with enzyme. Okay, let me go back to the biofuel. And so I just talk about the control group. And I just talk about why it's important to have a control group, right? So you can compare your you will have a baseline to compare it to, because you have baseline sugar, from all these plant biomass, and because of these baseline sugar, you will have baseline fermentation. So you really want to know whether these hydrolysis, how much more does it give you as far as glucose and alcohol is concerned when you added the enzymes. Now you remember in question 5 in this lab, the step is you go through pre-treatment, you boil it first, and then you have your enzyme digestion and then fermentation. So at which one of these steps you expect the most glucose, right? And then you say, let's say you think it's the first step, I don't know. And then you say why. You tell me why. Because you always explain. You think which step will generate the most glucose? Why? So it's one of these three. And then you also need to tell me whether you think it's in the control group or in the experimental group that has the most glucose. Same thing, asking about alcohol, right? Which step will have the highest alcohol? In what group? You can skip number 7. Now this table is basically a summary of all these questions that you have, from the answers that you have for these questions. So what you have here is, of course, you write down your research question, your hypothesis, right? You should have that already. Now control variables, remember you have to write down what are the 3 things that will be the same. You need to go back and read the lab, see what can you pick as the 3 things that are the same. Now, dependent variable is what is going to change based on the independent. For example, in my case, I have corn, cob, and hop. So my independent will be the 2 different biomass, corn, cob, and husk, right? So you have this. And then what is your dependent variable? Every group will be the same. Will be your glucose level, your alcohol level, right? This depend on this. So here's your experimental group. You have your biomass plus, in my case, because I pick option 1, it'll be my biomass plus enzymes. Control group will be biomass with no enzymes, right? Yes, Misty, control group is exactly identical as the experimental group. You have the buffer, you have the biomass, except you just don't add enzyme. You go through the same boiling treatment and all the temperature incubation. So we keep everything the same. So that could be your control variables. You can say you use the same buffer, same amount of buffer, you go through the same temperature treatment, you use the same amount of biomass. Can you see that now? So everything is the same in control except there's no enzyme. So prediction, like I said, will be similar, but you can have it in more detail. You can say have more glucose, so it can have more alcohol. Um, as long as it's coherent with your overall experiment design. So this table is this. You don't have to do a flow chart because it's not a wet lab, so forget about this. Now, once you finish this first 1 page, 3 pages of questions, 2, 3 here, right? 1, 2, 3. Then you email it to me as a Word file. Then I will email you this table with all the data. Okay. And once you have this table from me, I usually will, if you email me, I'll email you back within the day or the next day, depends on when you send it to me, I'll email you this table with data, completely fill out based on the option you pick. Then you're gonna do two graphs. Okay, one graph, I'll show you an example later on. One graph, you will show me the glucose level. It's gonna be a line graph. One graph, you're gonna show me the alcohol level. And those are the treatments. So on the graph, you will have, let me show you here now, so I can draw again, new slide. Sorry, I'm running a little over, but we'll be done soon. Okay, so on the graph, what you need is like this. So one graph is for glucose level, one graph is for, so the glucose, let's say this is glucose. And this is alcohol. So on the y-axis, you have your glucose number, and which is in, so this is glucose here. You look at the units, okay, it's on the table. And on this here, you have your pre-treatment here. You have your hydrolysis or enzyme digestion. You have your fermentation. Only 3 points here, okay? So your graph will look something like this, you know, and this one, same thing, 3 points here. You have your alcohol on this side. So this treatment is here, pre-treatment, hydrolysis, fermentation. Your alcohol level is here, so it might look something like this. Okay, so it's going to be a line graph. You need two graphs. Okay, you can attach that, or you can just send me your Excel, whatever you want to do. So those are the two graphs. Um, So I give you, remember you send me the first 3 pages, which is the experimental design. If you're done with it, I grade it, make sure you're correct. I'll send you this table, and then you use this table to make those two graphs I just show you. Okay, and then you use that to answer these questions. Any changes in glucose after enzyme digestion? Based on that data, you can answer this question. Answer this question. All these questions are based on the data table that I will send you. And then, of course, did it support your hypothesis, right? Now, we are not going to do these two questions. These two questions, because it's based on class day data. We don't have. So what I will do, I'll update the lab today to delete all the questions that you don't need to do. Okay. So you don't have to do number 1 or 2, but you do number 3 and 4. Does that make sense? Now, please ask questions now. This is the biolab stuff, biofuel labs. That's what you're supposed to do. I'm gonna go ahead and delete some stuff. No, and you don't have to. You just send the first 3 pages together. There's no due date, like I said, but you need to. Everything is still. So if you send, you have to send me those design the first 3 pages at least a week before the final exam, so I can give you those data. Just remember, once you send me those first 3 pages, I will give you the data, and you still need time to graph and stuff like this. So you don't want to wait till May 11th to send me the first 3 pages, because that way I can send you the data, but still you don't have enough time to do your graph. So everything is still, especially the report, is still May 11th. So there's no, I don't have a separate due date for each part. So you just want to keep track to get your design done soon enough that you can get your data and then do your report in time. So what I'm gonna do now? Any other questions? I'm gonna load a new updated version of the lab onto the biofuel project, so then you can have it. Change. Okay, if there's no other questions, I'm done. Remember, every Monday there is a study session. You can ask me questions about your biofuels project or anything else. And final exams is going to be due on May 11th. Biofuels project is going to be due on the same day. So remember, you finish the first 3 pages and then you send it to me and I'll give you the data. Okay? Any last minute question? Okay, if there's no other question, let's see. Yes, Misty. So the report is actually the data sheet. So remember, it's this one. So the lab report is starting on the data table here. So you have your data table, which I'm going to give it to you, right? And then so it's the graphs and the 4 questions and the 3 questions here. and then the 2 questions in the discussion. So last 2 pages of the lab is the report. Yes, and there won't be any more live stream at all because I'm done with all my lecturing, right? But what I did was I did keep the photosynthesis stuff on D2L. So in case some of you get bored and want to know about photosynthesis, you can still read over the PowerPoint. You can do the soft chalk, but there's no due date. It doesn't count for anything. It's just for fun. If you really want to do it, you can still do it. That's why I left it up there. Otherwise, I would have deleted them already. But I figure some of you might be interested. If you go on to science, you might want to look in. I mean, you might want to look into it and do it so you have some background knowledge of it. But if you're going into nursing, I don't think you'll ever see photosynthesis in nursing. So you might not want to bother with it. Yep, so that's why I keep saying there's no live stream anymore. So if you have questions, you email me or you come to the study session on Mondays, and then we can have more interactive question and answer time. And I think you run into questions about the biofuels stuff that you're doing, and you need to ask. Don't assume that you know how to do, and just those are good points. Is I think it's worth 5% of your overall grade. So if you're making a 65, that 5% can help you pass the class. Okay, I think that's. I know it's too bad it's the last lecture and I don't get to see you guys face to face for one last time. But hopefully I see you guys again on campus next semester in fall. Okay, I'm gonna end